Okay, thanks everyone for your patience. We're just about ready to get going now. Everything looks set up. So it's my pleasure to welcome Jaime Iranzo to give a talk at the CAB, which is where he's a científico titular since last year, which is also where Jaime started his career as a PhD student, where he developed mathematical models and applied these to viruses and genetics. After his PhD here at the CAB, Jaime went to the National Institute, the National Health Institute in the US, where he worked in Washington D.C. for around 6 years. And in 2019, Jaime won the Ramon y Cajal Fellowship, and he brought that here to Madrid. Well, not here at the CAB, but here in Madrid. And since December last year, he's been a científico titular here at the CAB. So I'll let Jaime take it away, and thank you very much for agreeing to give a seminar.
Thank you for the presentation. And thank you, all of you, for coming here today, also those that are watching us via Zoom. I will start with a quick survey just to get an idea of the audience. So if you don't mind, could you raise your hand if you are familiar with the idea of evolution? Okay, almost everybody, that's great. What about network science? Yeah. So great. I see you have more of a biological audience than physicists. That's good. Ah, que no va la antena. Ok. Ah, pero tenía posible. Can I use the the whole side? What was I? Yeah. So I will try to make things easy. If at some point you get lost or you need some clarification, just let me know, and otherwise we will have time for questions in the end. So let's start. What do I do? What does my group do? Well, we study evolutionary systems. So that means that we study evolution in a vast diversity of systems. We study evolution of genomes, evolution of viruses, evolution of microorganisms and microbial populations. And for that, we combine 3 sources of tools and information. So we are using tools from complexity science, including game theory, mathematical modeling, network science that I will talk about today later. Then we also use tools from comparative genomics, so classical phylogenetics, sequence analysis, and so on. And as our main data source, we use public data from big international consortia, like the Human Microbiome Project or the Earth Microbiome Project. So by combining all these 3 sources of data and methods, we try to get a better understanding of how evolution works. If this looks broad, it's because it is broad. So I'm going to focus on 3 particular examples where I will show how network science can be helpful to study different aspects of evolution. My goal is not just to teach you something, it's also to show what kind of tools I use, what kind of things I'm interested in, because as Lee mentioned, I joined the Astrobiology Center a few months ago, so I'm going to stay here probably for a while. And I think it's good that you know the kind of things I do and the kind of things I can do, so we can establish collaborations in the future. So first I will talk about gene sharing networks and how can we use them to infer evolution. Then I will talk about epistasis networks and how they could affect evolution by canalizing evolutionary trajectories. And finally, I will talk about about ecological networks and the way that they affect evolution. Okay, let's start from the beginning. Let's start with a little personal story. So when I was little, I was fascinated by dinosaurs. I love dinosaurs. I think I'm not the only one, like many people do that. And that may be, who knows, one of the reasons why I ended up studying evolution. So I think pretty much most people agree that complex multicellular organisms can be fascinating. However, the truth is that simple prokaryotes dominate the world. Okay, so this idea of Jurassic Park is not true. Like if you go look around and sample the diversity around you, like the vast majority are microbes. For those of you who are familiarized with phylogenetic trees, I put this figure. So this is from a paper in 2017 where they explored the whole diversity of life for which there was available sequences. And what we have here in the red circle, all these are, sorry, now I'm using my wrong hand. So all these things in this circle are bacteria and archaea, unicellular prokaryotes. That's all. Eukaryotes are here, but still, most of these are still microscopic. Okay, if you wonder where we are, you see there are no mammals in this tree, no vertebrates, no chordates. We have to go back to opisthokonts, which are like somewhere over here. So we are inside that tiny branch of the tree. But that's for you, for those of you who are familiar with these representations. For the rest, let's start from the basics. So, you know, probably are familiar with the ideas of a tree of life, this idea that Darwin introduced from the very early times of the origins of a species. Concept is that all living beings come from a single origin, and that original organism started diversifying, and we can represent this diversification of life as a tree. This very beautiful image was started to be tested in the early 90s when Carl Huss had the idea of studying this. I hope I don't say it wrong. He was looking at ribosomal RNA or ribosomal proteins. RNA. So it's correct what I have there. He started looking at the ribosomal RNAs, and by comparing the sequences, he tried to reconstruct this tree-like structure of life. So basically, you have many sequences, you put together the two that are most similar to each other, then you cluster down with a third one, you know, it's some sort of a hierarchical clustering, and then you end up with a tree. And one of the groundbreaking discoveries of that approach was that suddenly when people have been considered bacteria all together as a single unit, split into big domains, bacteria and archaea, and they are as different, so different with each other as they are with eukaryotes. So he already found that we are sort of the minority there. And that looks great. We get a tree. It's like Darwin's idea, tree of life. We are all happy, except that that's only based on a single gene, single ribosomal RNA. And we have many genes in our genomes. So what happens if we do the same for all genes? Well, the things get more complicated. With something like this, looks more like a bush. And we find something like this because genes are not only vertically inherited to the offspring, genes can also be horizontally transferred. There are a series of mechanisms by which this can happen. That's also part of the research we do in the group, but I won't go over that today. So the idea, we have many genes. Genes have different histories because they can be horizontally transferred. Another question is, what happens with this concept of the tree of life? Should we just throw it away? It's not valid anymore, or it's still useful in some sense? So this was a heated debate for several years, I would say even decades, until, let's say, 10 years ago, when people started taking the question and systematically analyzing it instead of just discussing about it in a hypothetical way. So one of the nicest approaches, I think, was this one from a paper from 2013, where the authors built phylogenetic trees for a large number of genes that are shared by mostly all prokaryotes. So this is mostly for archaea and bacteria. I'm not including eukaryotes here. They build the trees, they overlap them on top of each other, and they got this. And the conclusion is that, yes, we can see something that looks like a tree. So there is some sort of what they call the forest of life is a tree in a statistical sense. A majority of the genes follow this trajectory. And then on top of that, we can see some horizontal transfer. So, okay, that's sort of a nice way to solve the question. And that's when I jumped into this story, because I was interested in studying viruses and viral evolution. And I was wondering, well, can we represent viral evolution as a tree? Like, how can we bring together the paradigm of the tree of life, which is sort of like an iconic view of evolution, with viruses? And here we have several problems. Like, first, viral genes evolve very quickly. So we can't really make phylogenetic trees. We don't have enough information. Okay? Building a tree is not just about comparing sequences. The sequences have to be similar enough so we can get information from them. Also, there are no universal genes. We don't have ribosomal RNA gene that we can use to at least have a one gene tree. We can do that for narrow viral groups, but not if we want to study deep phylogeny. So the idea that was developed together with Eugene Koonin and Mark Rupovich was to use networks instead of trees to try to understand how viruses evolve. So I will explain it later in more detail. First, I'm going to tell you how we make these networks and then how we interpret them. To build a network, we do the following. We start from a large number of viral genomes that are available in public databases. They were available at that time. This was a few years ago. For each genome, we take their genes and we identify the gene families. Okay. Genes that have like capsid proteins, polymerases, and so on. We can do that by sequence comparison. And then we represent them this way. So we have the viral genome, will be V1, to use this for people who are at home. Well, apparently I'm not very skilled with this electronic pointer, but the V1 will be the viral genome, and the A, B, and C will be different gene families. You can think, for example, one is a polymerase, a capsid protein, a terminase, whatever. Then we take more genomes, and we do the same. And we end up with something that is called a bipartite network. So it's a network that has two kinds of nodes. On the one side, we have the genomes. On the other side, we have the gene families. A genome is connected to all the gene families that are represented in that genome, and a gene family is represented to all the genomes that have that gene family. And now once we have this network, we can start doing things. So the bipartite network conserves precise information about the gene sharing patterns. I'm going to show you that here. This is the network for, on the top left corner is the network for all double-stranded DNA viruses, the gene sharing network. And here on the lower right side, we have that network, the gene sharing network for archaeal genes, which are a subset, archaeal viruses, sorry, which are a subset of that whole double-stranded DNA virusphere. From here, we can see, like in color, I represented the different genomes. The white lines are the edges, so they are connecting genomes to genes. And when you see that those white lines intersect, those are some of the main genes that are shared by many genomes. Let's try again. Let's see if I'm luckier now. No, sorry about that. So, some things that we can see here. First, if we look at the top left network, the overall DNA virus network, we see that the archaeal viruses, the ones in color, come in two groups. One of them, to the right, is fully integrated with the rest of viruses. They are similar to viruses that infect bacteria or even viruses that infect eukaryotes. But then we have the rest that are very disconnected from other viruses. Those are viruses that infect hyperthermophilic archaea. So we have some interesting piece of information already from here. It seems that hyperthermophilic archaeal viruses are different from the rest. They don't share as many genes. They are sort of disconnected. Now, we can look at the genes that are shared by many viral genomes. So here in the lower left column, we have those 4 genes in the middle that I put their names. One is the capsid protein, the terminase, and the portal protein are involved in the packaging of the viral genome inside the capsid. So these are genes that are important to make the viral particle. And it makes sense that they are shared by most of the viruses. On the right, on the left, for archaeal viruses, we only have a few genes that are shared by many viruses, like this RHH domain. If you know about the biology of viruses and of these organisms, these are not particularly hallmark viral genes. These are genes that are often present in organisms that live in these hyperthermophilic environments. So these were probably taken from the host in independent events. They are not telling us anything about those the origin of those viruses or how they are assembled. So that's another interesting piece of information. And from here, we can develop some hypotheses. The most interesting is that perhaps double-stranded DNA viruses that infect archaea, and especially hyperthermophilic archaea, have different independent origins from the rest. And also, it's telling us that perhaps horizontal gene transfer among viruses that infect this hyperthermophilic archaea is much lower. Why? We don't know. Maybe because of the structure of this environment, because it's much harder. The DNA is much less stable in this environment, so it's harder to transfer it. We don't know yet, but those are hypotheses that we get from the network that we would like to test in the future. Also, I want you to focus on the fact that when you look at the viral genomes and the color circles, they come normally in clusters. I put them in different colors to make it easier for you to see. These clusters have to be densely connected with each other. They share many genes, and they only share a few genes with genomes from other clusters. This is what in network science we call communities or modules or clusters. Okay, so it's groups of nodes, in this case, groups of viruses that share many interconnections with each other. In this case, they share many genes, and they don't share so many connections with genes that don't belong, with genomes with viruses that don't belong to that module. Here is the representation with just a generic network. On the right, you see a representation with in the virus gene sharing network. What does it mean from a biological perspective? Well, the logical interpretation is that viruses that belong to the same community are phylogenetically related. They share many genes, probably because of common ancestry. So we could, in principle, use these modules, these communities, to identify or to define viral taxa. We actually tested if this idea works with RNA viruses. This is with a network at the community level. So each node here is not a genome, it's a community, a group of genomes that share many genes. And then we check how that agrees with the traditional phylogeny traditional taxonomy of viruses. And there's quite good agreement somewhere between the genus and the family level. So it seems that actually we can use topological information from the network to infer taxonomical information. This is very useful, for example, if you discover a new virus, you go to the environment, you do some metagenomics, you assemble that, and you predict, okay, this is a viral genome, but I've never grown it in the lab, I don't know what it infects. You can use this strategy to try to assign a taxonomy to that virus. And this is not just an idea, it's like some people actually took over this proposal, and now some of the most popular tools for viral taxonomy, for automated viral classification, are based on this network approach. But we can go further than that. We can take this idea one level higher, because we can also define higher order modules. So sets of communities that are densely connected with each other but not with the rest. And as you can imagine, by applying this iteratively, we can generate a hierarchy of modules. And once you have a hierarchy of modules, we can get something that looks sort of like a tree. So we do this for the whole double-stranded DNA set of viruses. And I would say that perhaps the most suggesting finding of this work was that we can cluster most viral genomes in two big supermodules. Those two big supermodules are mostly separated by the kind of capsid proteins that they have and the packaging machinery. And each of those modules includes viruses that infect eukaryotes, prokaryotes, and eukaryotes, bacteria, and archaea. So it is possible that these two big groups of viruses developed before the diversification of life into the three domains. Right, so that's how we can infer ancient evolutionary relationships using these gene similarity networks. Okay. We'll go to the second topic, which is about epistasis networks and canalization. But I'm going to start with a little tour, like a little quiz to make you think a little bit. So now we have a black box. I'm going to give some properties about an organism or about some entity in the world. And we are trying to get information about that just based on these hints. Right? So we start with the following. We know that we are looking at something that can grow. We are looking at something that can produce copies of itself. It has well-established physical boundaries, and it has a metabolism. So what would we say about this? We are in the astrobiology center, so what would be the first conclusion you would take of this? Yeah? It's alive, it's a living being. Do we all agree about that? Like, if it's made of chemistry. Yeah, well, I try to convey that it has a metabolism, but maybe the thing is more subtle than that. Yeah, but okay, we can assume that it's a physical entity, it has chemistry, it's a living being. So far, so good. So now it's the time that we call some geneticists and let's do some genomics here. Let's take some samples, analyze it, and see if we find out what kind of living being is. And then we found out that it has the genome of a dog. So, okay, we are done. We are happy. It is a dog. Do you agree? Is it a dog? Is it necessarily a dog? I don't know if someone here knows about this story. I chose it for a reason. Because what I'm talking about is not a dog. It's what they call here cancer parasite. It's actually a lineage of cells that developed more than 6,000 years ago. It was a tumor that became infective. Now it can pass from dog to dog, yes, through sexually transmitted disease. So this thing has generations, has been stable for 6,000 years. The genome is quite stable. It comes from, it's a dog genome that has, of course, has mutations and reorganizations, like all cancers do, but now it's infective and it's a thing that replicates on its own. So what is it? Is it a new species evolved from a dog? Should we call it cancer just because it originated as a tumor, but now it's an infection? Actually, it's an infectious agent. So what is it, and how does it challenge our ideas about how evolution works? Like, are we studying something that evolved from a dog? What's it? And the reason why I think this is hard to answer, or at least a bit puzzling in the beginning, is because it challenges some of our usual assumptions about how evolution works, in most cases. It actually connects with an interesting topic that you may be familiar of, because you are where you are, which is the idea of major transitions in. Oh, sorry, before going to that, I tried to put a photo so you could see how it looks like, but this is what Google returned. Obviously, it's all photos of dog genitalia that for some reason Google isn't very happy about. So I don't want to be censored, I didn't put them. Now, major transitions in evolution. Could you raise your hand if you are familiar with this book or with these ideas? Okay, not many, so I will explain it. So, this is from a book, like some years ago. It's a very popular book in evolution. I really suggest that if you have some free time and you're going to read something interesting in evolution, you could take a look. It's an outreach book, so if you have a biological background, it's going to be super easy for you. The idea is that most of the major transitions in the history of life, from the origin of life to the origin of language, can be explained from a conceptual perspective as the result of the interaction of entities generating a higher order entity that ended up being a superior level of selection. Okay, so it's how these layers of complexity are formed during evolution. The thing is that there are also reverse transitions. We are used to thinking of these like smaller things building bigger pieces and more complex, but sometimes the opposite happens. And cancer is one of those examples where you have a multicellular organisms that revert to a unicellular level of selection. So cells start competing with each other inside a tissue to replicate faster, and that's what actually is cancer. And in some rare cases, as these transmissible cancers, there is another example in Tasman demons, become organisms themselves that can actually replicate and perpetuate over time. I felt obliged to put this because this is a zoology center and I'm going to talk about cancer. And you could ask, what is this guy talking about cancer here? So I want to show that cancer can bring up relevant questions and deep questions in the context of evolution. Cancer is also a great model to study evolution. People normally think of viruses, and it's true because they evolve fast. You have large numbers in the laboratory. But in the case of cancer, first, we have hundreds or thousands of replicates. It happens all the time. There are a lot of available data because it's something of clinical relevance and people have spent a lot of money sequencing tumors. And those are independent replicates, so that's great. We have independent evolutionary histories, then we can do statistics with that. Moreover, cancer cells are subject to strong selection pressures. They are out of equilibrium, so probably we are going to see adaptive evolution there. And just more suggestive or speculative points, they could be also a good model to study these reverse transitions in evolution, which kind of forces keep high-level organisms together. What happens when these forces disappear? Why does complex life, why is it stable and it doesn't destabilize into the lower levels of simpler replicators? And finally, this is highly speculative, but there is a paper about that, so included here. Some people have suggested that cancer could be a model for a minimal replicating construct. Basically, everything is screwed except the basic mechanisms that a cell requires for replication. So they suggested that we could learn something about a minimal cell by studying cancer cells. Okay, so I hope I convinced you that studying cancer may look exotic in a place like this, but still it's worth just spending some minutes talking about it. Now, let's talk about the main concept of cancer evolution. So, yes.
How well established is already the idea that cancer can be used as an evolutionary model?
Not a lot. Like some people are convinced about that. It's funny that you ask this because when I was doing the exam for the scientific titular position, I had that slide and I removed it in the end. And one of the members of the committee was asking, why didn't you go deeper about that? Like, I think cancer is a great model to study evolution, and people normally are not aware of that. I would say people are not aware of that. Even people working in cancer normally don't think about that as an evolutionary process until quite recently. So in the last maybe 5-10 years, there are more and more, you can start seeing in papers and reviews from people working in cancer where they are applying evolutionary genomics to study cancer. They are doing phylogeny of cancer clones. So it's starting to permeate in the field of cancer that looking at it from an evolutionary perspective can give useful information. I don't know if it's so well accepted by people working in evolution that they could use cancer to learn things about evolution. That other side, I don't have a feeling that many people are using it as an evolutionary model. And perhaps that's why I thought that it was worth mentioning it here. Okay. So cancer basically, is a population of cells. They acquire some mutations. That mutations allow those cells to proliferate and escape the control mechanisms of the tissue. We call those mutations drivers. Okay? On top of that, cancer cells accumulate other mutations that we call passengers that are neutral or even slightly deleterious, but they just happen to be there and they take advantage of the neighboring driver mutations, so they proliferate with the cell population, with the rest of the genome. Another big question here is, as I mentioned before, every tumor in different people are independent evolutionary processes. So why is it that we actually see regularities, that we can classify them into subtypes that are sort of recurrent? Like, what makes these independent processes converge to stable subtypes, to stable phenotypes? I think this is a quite interesting and general question, not just about cancer, it's about evolution in general. So for that, I will first mention a concept that is the idea of epistasis. Maybe many of you already know, but just in case. The idea is that mutations, and in the case of cancer, driver mutations, don't act together, don't act alone, sorry. It's well established, for example, that for some cancers to develop, you need to have a mutation in two genes, like this NYC and KRAS. If you only have mutations in one of them, nothing happens. When you have mutations in the second gene, then cells start proliferating. This is a particular example of a general phenomenon that I mentioned before. It's called epistasis. Epistasis means that the effect of a mutation depends on which other mutations are present. What happened before? Okay. This is often represented as fitness landscapes, as RAC fitness landscapes. I won't go into much detail over there. But what that means in the context of cancer is that selection for a mutation to proliferate and for that clone to grow is dependent on, or is conditional on, what mutations happened before. Right? So for example, here, if we have first a mutation in A, nothing happens. This plot here will represent the fitness, so the fitness is the same. Those cells won't replicate faster. If you have a mutation in B after that, then that clone is heavily selected. But if those cells with the first mutation in A weren't selected, then maybe yes, that clone, that never happens. The other trajectory going first through mutations in B and then you have a mutation in A is much more frequent because the first clone is selected for, because it has a selective advantage. So the end is the same, but the paths are not equally likely. And what we see here is that there is this conditional selection. So selection for mutation A is conditional on having a mutation in B. We can leverage this idea, apply some statistics, and use that to infer the epistasis networks. So again, these are technical details. There is a software that we developed for that. We apply it to cancer, but it can in principle be applied to any system like bacteria, viruses that you have, as long as you have enough independent replicates. It's basically applying advanced statistical inference plus concept of conditional probability. Once we have that, we can build what we call the epistasis network. So it's a network where each node represents one gene. Two genes are connected if selection on one is conditional on having mutations on the other. So if there is epistasis. Here I'm represented in green cases of positive epistasis, so mutations in A promote or increase the effect of mutations in B, and in pink, negative epistasis. So when you have mutations in one gene, that sort of cancels the effect of mutations in the other gene. So they become mutually exclusive. On the left, you have a generic network. On the right, you have a real case for a kind of brain cancer. So the genes have names. They are well-known cancer genes for that tumor. And we see that this network has some structure. I'm going to go into that with more detail. So there are groups of genes that tend to be connected with each other through positive epistasis, through synergy, and separated from the other genes through negative epistasis. Does it remind you of something? This is the same idea of communities that we saw before for the viruses, but now, instead of having no connection between two different communities, what you have is negative interconnections between two different communities. So it's this sort of sign-based modularity. So we'll have a first community here with these three genes that tend to synergize with each other. We have another community there, and a third community down there, and then these two nodes that go on their own. If we study, we can go to a particular patient, look at which mutations that patient have, and classify that into one of these communities. If we do that, and then we study survival, what we find is that survival depends on which community of genes is mutated on that patient. So this is important. This isn't just like we are learning something basic about basic science of cancer. This has applications. This can give us information about how malignant that tumor is. This is another case for endometrial cancer and colon cancer. So again, we have the same structure. Genes that are connected through positive epistasis that are synergistic, and they are negatively interacting with other sets of genes. And if we look at these genes and we ask an oncologist to give us our opinion about that, it's like, okay, what you are looking at are genes that are typically mutated in different subtypes. So this will be the copy number high subtype of endometrial cancer. This will be the endometrioid type 1 cancer. And for the colon cancer, it happens the same. So this will be the canonical pathway to colon cancer, has been very well studied. And this will be the consensus molecular subtype 1. So there seems to be a correlation between the structure of the epistasis network and the subtypes that different cancers that are independent to each other converge to. So the hypothesis is very, very clear. We will think that subtypes like well-differentiated subtypes, consistent subtypes, are the result of this sign modular structure of the epistasis network. To test that, we build a model. We did some simulation. So in our model, we start from different kinds of epistasis networks. Then we simulate a population of cells. Those cells grow, and they proliferate more or less depending on the combination of mutations and if they are connected through positive epistasis or negative epistasis. In the end, we get a mutational matrix. We get our population, and we see which mutations we find in each cell. And then we just cluster those mutations using a typical like principal component analysis or whatever to clusterize them, to try to find tumors that are similar to each other. And what we found is that when we have epistasis networks that look like the ones in reality with these communities of genes connected through positive interactions, then we can find tumors that cluster together in well-differentiated subgroups. So it will be these three ones in the middle. So it seems that actually tumor subtypes emerge from this canalization of the cancer evolutionary process that is driven by the structure of the epistasis networks. We went a bit further with this work. So we studied, actually, Carla Alejandre, who is here now, studied how this positive epistasis affects cancer evolution. And she found that there is a connection between cancer evolution with epistasis and a phenomenon that is very well described in ecology, which is called evolutionary rescue. So when the environment changes, population can go extinct unless something happens, a mutation happens that rescue that through evolutionary changes. And there is a connection between that and the effect of positive epistasis in cancer. It is informative because it is telling us that if we want to try to predict how an early tumor lesion is going to go, we have to focus on some particular genes of the epistasis network, and we know which one we have to look at. But it is also suggesting that maybe when we study evolutionary rescue in an ecological context, maybe looking at epistasis should be important. So these are some of the summary conclusions of this part of the project or of the talk. I would highlight the second, epistasis is pervasive in cancer genomes. These networks are sign modular, so they have high modularity. And finally, the structure of these networks could be the reason why cancer populations are canalized through these well-established subtypes. Then I will finish with a part about ecological networks. So for that, I want to introduce two concepts that have something in common. So first concept is pangenome. It's the set of all the genes that are contained by all the members of a species. So if you think of human genome, more or less all the humans have the same genes. If you look at bacteria, that's not the case. You take two genomes of E. coli, and many of the genes are unique from one genome or the other. Or whatever your favorite bacteria is, you took several isolates of that population and you will find that their genomes have very different gene contents. Some of them are shared by all the members, that's called the core genome. Some of them are unique from a single strain or just from a few strains. So the whole set of that is called pangenome. And then I will talk about metagenome. Metagenome could be thought of as the collective repertoire of metabolic functions and other functions that are present in a microbial community. In these cases, the genomes are probably in individuals from different species. But still, if the gene products can be shared or the metabolic products can be shared, we can think of the metagenome as some sort of collective genomic allocation. So I think this is an interesting way to look at microbial evolution. Hasn't been very deeply explored so far. The idea that in microbial evolution, we have a lot of collective gene allocation or distributed gene allocation. You don't need to have all the genes in the same genome for them to provide benefits to the community as a whole or to the species as a whole. This connects with an idea that was developed by Lenski's group based on his famous long-term evolution experiments, which is called the Black Queen hypothesis. Not to confuse with the Red Queen hypothesis, that is also a very interesting thing, but it's a different thing. So the Black Queen is based on this card game that became popular with computers, where it is that you have to get rid of as many cards as you can. So the way they put it in biological terms is that when you have a biological community that is stable for long enough times, the members of that community become specialized metabolically. So you don't produce, you don't perform a function if someone else in the community can do it for you. And the result of that is that different members of the community start losing genes and metabolic pathways, and they develop a very interconnected network of metabolic interdependencies. So I get rid of everything that I don't need because someone else provides that for me. That's the hypothesis. We want to test it. Is it true that actually microbial communities are highly interdependent from a metabolic perspective? And if it is true, how does it affect the dynamics of those communities and the long-term evolution of those communities? So to do that, we started a project with a main goal, which was to characterize these cooperative metabolic interactions, in particular in the gut microbiome, because we have a lot of data for that. And for this, it's important to have very good data and very good quality, as you will see. So very quickly, through the pipeline, we have the genome, we have the community composition. From that community composition, we can reconstruct the metabolic model. So what are the metabolic capabilities of a particular species or a particular strain, and then we put all them together. We assume some ability to share metabolic products, and then we can infer this metabolic interaction. So we can see, for example, if species A can produce something that the species B needs to grow, or if they can share things bidirectionally and then they became mutualistic. So yes, going again, we have this metabolic model. We have a software called Smetana. It wasn't developed by us. We are just using it. And then we can infer this kind of interaction. So we can have a species that are self-sufficient for growth. Given a medium, we have to define the medium, what nutrients are available outside. Some bacteria can grow on their own. Some others can grow because they take nutrients from another bacteria. So that's classical commensalism in ecology. We can have mutualism. In this context, it's called cross-feeding. So one bacteria provides something to another one and vice versa. And that's pairwise interaction, the classical interaction from ecology. Other two. We are looking at higher order interaction. What's that? Those are interactions that require more than two species to work. So in this case, for example, order 4 interaction to the right means that one bacteria requires nutrients produced by 3 different species. So that can only grow if the 3 other species are present. That's an order 4 interaction. It involves 4 species, and we can't, if we remove any of those, that interaction doesn't work anymore. Or we can have order 4 mutualism. So we have this kind of autocatalytic cycle where each bacteria depends on the previous one and they close the cycle. And if you remove any of those, that doesn't work. So that's order 4. We can't represent that as a square with 4 pairwise interaction. It's not like that. You have to have the 4 species at the same time. To stress this point, an order 4 interaction is not the same as 3 order 2 interactions. Okay. In this case, for example, the lower right case, like any of those 3 bacteria could help our focal bacteria to grow. In an order 4, we need the 3 of them at the same time. The same with the upper example. So, is it clear what a higher order interaction is? These are the kind of complex interactions that the Black Queen hypothesis predicts. So, we have the data, we have our methods, we can actually quantify these kind of interactions. We use different media because, as I told you, it depends on how many nutrients you have in the environment. If you have all the nutrients, then you don't need to interact. You can get everything from the ambient, from the environment. If the environment is very poor, you really need to establish interaction. So it's important to try different environments. We also try different methods to infer these metabolic networks. So I put it as if it is easy, but it is not. This inference of metabolic capabilities has a lot of uncertainty. There are different methods, there are different approaches, and it's important to test how robust the findings are with respect to them. The first thing is that, well, this tool doesn't work very well. That's not good news because this is the method that we will use if we want to study bacteria from poorly characterized environments for which we don't have curated metabolic models. So sad news because it means that for the gut microbiome, this is good because the bacteria have been very well studied. People have spent a lot of time trying to make highly curated metabolic models that work. But if we want to apply this method for, very strange or unusual environment where the metabolism hasn't been very well curated, it's not going to work. That's not good. But other than that, if we look at the distribution of the interaction order, we see that there is generally a peak around 4. So that means that 4 order interactions involving 4 species simultaneously are dominant in the gut microbiome. Now, what kind of interactions we have? Well, a little bit of everything. So we see further or commensalism, the thing on the top, where one species requires over 3 other species to grow. We see strict mutualism, this kind of autocatalytic cycles that require like 4 species. And we also find, and it's actually dominant, more complicated, more exotic configuration like this one, low order mutualism plus commensalism. So one species is commensal or on a set of mutualistic bacteria. Here in this case, we have 3 bacteria that have a mutualistic cycle, and then the 4th one depends on that cycle. So it's not just we have higher order interaction, it's that we try to simplify things and make them look like, okay, commensalism, mutualism, and that's all. And sometimes when we go into high order, the most relevant interactions are quite more complicated than that. They don't fall into our classical categories of ecological interactions. We can also build ecological network and find keystone species. Some of them have been described as being important for having a healthy gut microbiome. So it makes a lot of sense because they are providing services, they are providing metabolites to the community. We can also use this to detect auxotrophs, so species that require other species to be present to grow. So what are the implications of this? Like, okay, we know there are higher order interactions, but can we just forget about that because it's too complicated? Can we just assume that, we are happy with our typical pairwise ecological interactions? Well, the answer is no. I don't have much time to go over it. It's also quite technical. Here is where my first equation appears in the presentation. But the thing is that we did some models to study how these high order interactions affect the stability and the diversity of the microbiome. And what we find, I will go here, for example, is that when you have high order interactions, what you get is opposite to one of the most classical and most, accepted axioms in ecology, which is that there is a higher limit to biodiversity. So the classical ecological theory for macroscopic ecosystems is that as you include more and more species, you will get to a point where the system just becomes unstable and collapses. You can't add species and indefinitely to an ecosystem. What we found is that when you include higher order interactions, the situation reverts. And the more species you put, the more stable your ecosystem becomes. And that would explain, for example, how we have so many species in the gut microbiome, which is one of the big questions of microbial ecology in the context of the gut microbiome. We also found that these higher order interactions can stabilize communities that otherwise would be unstable. If you just look at the order 2 network, like the classical network of interactions, and you put it into a model, you say this shouldn't be able to grow, this should collapse. If you include this background of higher order interactions, those things can become stable, and then you get much more diversity. So we think that even if it is complicated and we would like to simplify things and forget about them, we can't, because this completely changes the game. And with this, I want to finish just thanking the people who have participated in these projects. So the first part about the viral network was done while I was a postdoc at Eugene Green's group, in collaboration with people from his group and also with Mark Rupovich. Then the part about cancer evolution was done by a former student, George Grunhagen, who is now at Georgia Tech, and Jorge Cayo, here in Roy in the photo, and Carla Alejandre, here on the right side. She's in the room now. And the final part about the metabolic interactions is mostly the work of Adrián Aguirre, who is now in Graz in Austria, and also Adrián López, who is doing a PhD. He's still at the CBGP, like part of my former research group. So that's all for now. I'm really happy to take questions. Thank you for your attention.
Thank you very much, Jaime, for the great talk, and I'd sort of particularly appreciate you sort of starting, at the basics at the start, given the multidisciplinary nature of the institute. So, if anyone wants to ask any questions in the room, please raise your hand. Victor.
Sí. Thank you, Jaime, for the talks. And I see a lot of always bioinformatics. Always for me, sometimes I don't know if believe it or not. I mean, I believe you for sure, but our class, but everything for sure is must come from the sequence, come from theoretical connections. Because, for example, this ecological interaction that you showed us in the last part, I guess that this come from the comparison of all the sequences that you.
Yeah, these are predictions.
Yeah, you somehow understand that the metabolism of this group may complement this one or not, but for sure, just theoretical. But because you at the end you don't know what is really happening. But what is interesting here is that, as you said, the higher the number of the interaction, the more stable the population. This is interesting. I just try to think in astrobiology perspective. In just imagine a planet where there are only, for whatever reason, there are only 2, 3, 4 initial species, and for whatever reason, it was impossible to get a higher diversity. What do you think about that? Is this could be a reason for extinction, for example?
Not necessarily. I think this answers the opposite question, which is how is it possible to have super rich ecosystems with a lot of species and keep them stable? If the number of species is low, what this modeling framework shows, like typically the classical approach by May says that mutualism or positive interactions are bad. And there is even a paper about that for the gut microbiome. They say like the reason why the gut microbiome is stable is because most interactions are competitive. And that doesn't go very well with our regular experience of bacteria establishing consortia, like in biofilms or where you see mostly cooperation. There is, of course, competition, but we see a lot of cooperation too. What this is telling us is that when you take into account higher order interactions, when you take into account something that I say here, which is active growth, like ecosystems that have biomass input and output, what we found is that positive interactions, like cooperative interactions, become stabilizing instead of destabilizing. So if these species cooperate with each other, the system becomes much more stable. And that sometimes that requires these higher order interactions. So if we try to simplify the picture and just see like species A cooperates with B, B cooperates with C, but we forget that they make a triangle and the triangle is closed, then we can get to the wrong predictions. So that will be my qualitative take. Like to understand why small systems of mostly cooperative species are stable, we have to take into account that maybe these cooperative interactions are dependent on the presence of a third species. We have to look at the whole thing, not just at pairs of species. Regarding your first comment, you're totally right. So these are predictions. We tried many different methods to check that they are robust, but now what will be the breakthrough here will be to show this experimentally, to be able to develop a system where we can change the strength of interactions and also the order of the interactions and show that this actually is true. And I would really like to do that, but for that we need maybe an ERC or some serious funding.
That was my next question. Just try to put into the test tubes some of these ideas, because for example, when you talk about the genes, different genes, you always make some you draw the arrows and different length, different you know, how did you get that conclusion because it's not that easy. Okay, why this arrow is bigger than this other one?
Yeah, well, in the particular case of the cancer thing, that's something we get from the data. So if we see that a mutation is always present, it's only present if another mutation is present, we know that the interaction must be very strong because it's
statistical data.
It's a statistic, that one is statistical data.
There are some certain number of works that has previously identified certain.
Yes. In that particular case, is data from the what's the name? It's like the biggest data set on cancer genomics. We are now, well, the project is a bit stopped. We started trying to test this looking at different times, because what we are making is a prediction about how cancer evolution proceeds. We are basically saying that the first driver mutation is going to determine which evolutionary pathway that tumor is going to have. I didn't say that explicitly, but that's what this is saying. If you are able to identify the first driver mutation, you will be able to predict if the cancer is going to work towards a given subtype that is very malignant or another subtype that is not so malignant. And to test that, we need to have time-resolved data. And I started a project with a collaborator to try to look that from mouse cancer models, but we don't have results yet. So and I want to stress that we applied this to cancer because we had that data set that was very good, and also because the student who started the project was interested in studying cancer. But in principle, that could be analyzed to any experimental evolution data set with bacteria, with viruses. We could try to apply that to Lenski's data set and see what we get. So that's another direction for future expansion of this work into other domains of life.
Thank you.
Okay, well, I think we've got time for another couple of questions.
Thank you very much, Jaime. Super interesting. You said that it is possible to know the key species of the human microbiome, but it is possible to know the key compounds to maintain the functionality of the microbiome, or how important is a certain metabolite?
So first, that's going to depend on the environment of which metabolites are present, which in the case of the human gut is especially complicated because that changes. Even it changes during fasting periods and eating periods. But if we are able to define that environment, we can do it. Also, we can get some sort of time average of what happens overall. Or if we can see that a kind of nutrients is always or is mostly always involved in the interactions, regardless of the conditions, we say, okay, this is robust. So in theory, that's doable. In practice, that's the only reason that is keeping us from publishing this paper or finishing the manuscript, because we are having some technical problems with the software we are using to predict that. But it should be solvable, and we expect to solve it in the next weeks, hopefully.
Okay, thank you.
Thanks, Jaime, for this fascinating talk. I have a very easy question and another one that is not so easy. Both of them are very short. The first one is, how dependent are your results on the different algorithms used, in particular those relating to the finding of communities, which you know that there are lots of different algorithms and you have to tune them?
It is tricky. You have to use powerful algorithms because these networks have a lot of cross links. First, something that I didn't mention, it's more technical. We actually filter part of the links to keep the ones that we think have most evolutionary information. So those that there is like a background of things that are shared all the time, like integrases, defense systems, those are exchanged very quickly. And if you add them, they add a lot of noise to the network. So we implemented an algorithm to filter those out and keep the genes that have more evolutionary information. And we can discuss about that later if you want. And then once you have that, there are models that you can recover clearly, no matter which tool you use. And then there are some models that are more blurry. And for this, for example, we're using a consensus clustering approach. So we're running the same clustering algorithm many times, and then we try to find the consensus. If you do that, basically, the key is to use this kind of consensus approaches. If you use this, it doesn't really matter which. We try with different methods for community detection. They provide the same result as long as you don't just rely on a single realization. You do many, and then you find this you run this consensus approach, and then what you get is quite robust. But
and the second question, which is much broader, I would like to know your opinion on. Okay, we have been able to find sequencing that all the living organisms are come from a common ancestor. If you use network theory as you're doing to viruses, to all of them, how far can we get? How much information can we get from their origin? Not only their evolution, but even their origin. When did they appear? And are they really? That would be one question. And the other one is, how far can you get information from their relationships with bacteria, for example? Are there any kind of connections there in the genome? So can you see these connections there? And so which is your opinion of how far you're going to get with this new point of view?
So, for example, I think that the deepest prediction we can do in terms of going back in time and is this idea that I mentioned, that these two large, we call them super domains or super modules of viruses, predate the diversification of bacteria and archaea. Because we are pretty convinced that these two super modules represent true separated components of the viral world. We see a lot of gene exchange between members of the two groups, but we haven't found a single case where one of the main hallmark genes, the ones that make the viral particle, the capsid protein, the termin, we haven't seen a single case of gene exchange between these two groups.
Okay.
Between these two groups in those genes, so that makes, and these genes make sense because they are making the viral particles. So we believe that this is meaningful, and then we see that inside each of the groups we see viruses that infect archaea, bacteria, and eukarya. And when you look at those groups, those different viruses infecting 3 domains of life, it doesn't really look like one's evolved from the other afterwards. We could be wrong because viral evolution is very fast, but actually what it looks like when you look at that is that actually this group of viruses existed before the diversification of bacteria, and then they have diversified with their hosts. I think this is perhaps one of the deepest things we can say about viral evolution. Then, of course, there is groups like Mark Rupovich, for example, doing a lot of work on studying the origin of different viral proteins, try to guess which ones have been evolved from cellular components and which ones could represent ancestral genes from the cellular gene pool. This is difficult and there is controversy around it. So but we can talk about that later. But this is not the kind of things I would do with networks. They are using very advanced structural prediction models and other kind of techniques.
Right. Thanks a lot.
Well, thank you very much, Jaime. Once again, that's all we've got time for today. So if you'd like to thank Jaime again, that'd be fantastic.